environmental control, which is stromal cell mediated hematopoiesis control from the bone marrow environment, or the gene control apoptosis. Apoptosis is a condition which is known as programmed cell death, or growth factor, which is humoral regulation that regulates the hematopoiesis. Any disturbance of any of these three components can lead to a disease. What are the number of growth factors that regulate and act on hematopoiesis? These growth factors include cytokines like interleukin. We have several kinds of interleukin that act to stimulate hematopoiesis. We have interleukin 1, interleukin 2, interleukin 3, interleukin 4, interleukin 5, interleukin 6. Also, tumor necrosis factors act on hematopoiesis and stimulate hematopoiesis. We have growth factor like granulocyte colony stimulating factor and granulocyte monocyte colony stimulating factor, and we have hormones like erythropoietin and thrombopoietin. Erythropoietin stimulates erythropoiesis. Thrombopoietin is trigger this stimulate erythropoiesis and formation of RBCs. Thrombopoietin is trigger the formation and stimulate production of platelet, megakaryopoiesis and platelet formation. Interleukin 3 is pan-hematopoietic growth factor. This means that it stimulates all kinds of cells release. So this is a normal pathway again. Pluripotent stem cells give rise either to lymphoid stem cells or myeloid stem cells. Upon specific growth factors, it can give rise to specialized mature cells. For example, eosinophil need interleukin 5 for their differentiation and maturation. Basophil need interleukin 4. This is a very nice diagram about the myelopoiesis, in which is this pluripotent hematopoietic stem cells. It is present normally. The hematopoietic stem cells are present in stage 0. Stage 0 means that a quiescent stage. It is a non-dividing stage. But when there is a stimulus and there is division, it goes to stage G1. This happens by several factors like stem cell factor, interleukin 1, interleukin 3, and granulocyte colony stimulating factor or interleukin 6. And it committed to this is colony forming unit, granulocyte, erythrocyte, megakaryocytes, monocytes, which are under specific growth factors. It go to a specialized cell type. If it's exposed to interleukin 5, it gives us eosinophil. If it have exposed to interleukin 4, it gives us basophil. If there is thrombopoietin, it gives us platelet. If there is interleukin 3 and granulocyte macrophage colony stimulating factor or monocyte colony stimulating factor, it gives rise to monocytes. By the way, in the peripheral circulation, monocyte, if it enter in the tissue, it become macrophage. If this colony forming unit, granulocyte, erythrocyte, monocyte, megakaryocyte are exposed to thrombopoietin, erythropoietin, which is released in response to hypoxia and as in condition of anemia or hemorrhage or high altitude, and the release of erythropoietin from the kidney, it stimulates erythropoiesis, and this cell will be commit to produce RBCs. This is the terminology of the hematopoietic progenitor. This is the abbreviation. Colony forming unit, granulocyte, monocyte. This is colony forming unit eosinophil, and this is colony forming unit baso, which is mean the basophil. Colony forming unit meg, which means that it is the precursor of megakaryocytes, and this is the precursors of the erythrocyte is called Persa forming unit, erythroids. Okay. So the hematopoietic growth factors is a family of hormones or cytokines. It regulates the proliferation, differentiation, maturation of progenitor cell. It also regulates and affects survival and function of different hematopoietic mature cells. Hematopoietic growth factors, they are glycoprotein in nature. They bind, how they act? They bind to specific receptor on the progenitor and commit the progenitor cell to the specialized cell, kind of cell that the body need on demand. Okay. What are the characters of this growth factor? They are naturally occurring hormones, and they are low molecular weight and variable degree of specificity. As I mentioned, we have interleukin-3, which is pan-hematopoietic growth factor. It can stimulate all kinds of the blood component and blood elements that we know. Okay. I mentioned this before. Erythropoietin, it stimulates erythropoiesis, and thrombopoietin stimulates megakaryopoiesis. Cytokines are produced from different kinds of cells. It produced from monocytes, lymphocytes, fibroblasts, endothelial cells, and it acts on several targets. One of their targets are hematopoietic cell. It also acts on liver cell, endocrine, and lymphoid cells. As I mentioned, cytokine family, it can be interferon, interleukins. The one which I need here and we discuss here is interleukin 1, 2, 3, 4, 5, and 6, which are I need during hematopoiesis. They have several different family of cytokines which are involved in inflammatory response and immune response. Endothelial cell also can secrete, and this is a different source of growth factor, like granulocyte, monocyte, colony-stimulating factors, and the granulocyte, colony-stimulating factor are released from the endothelial cells. Some other growth factors are released from macrophage, and some released from the T cells and from the fibroblast. This slides for the master degree, not for the undergraduate. Also, this is for the master degree, the cellular cytokine production. Okay, natural killer cell produce granulocyte, monocyte, colony-stimulating factor, and also the P cell produce granulocyte, monocyte, colony-stimulating factor. It produces also interleukin 2, interleukin 5, interleukin 6. The marostroma provide environment for these growth factors and for the stem cells to divide and proliferate and mature into viable cells. It provides the stem cells with granulocyte, monocyte, colony-stimulating factor, granulocyte, colony-stimulating factor, and monocyte, colony-stimulating factor. It have also interleukin 6, interleukin 3, stem cell factor, and interleukin 11. The renal parenchyma is important for us because it provides with erythropoietin. Erythropoietin is the magic, or is the pacemaker that control the erythropoiesis. In response to hypoxia, the renal produces this erythropoietin, which is going to stimulate erythropoiesis. So the control mechanism of hematopoiesis depends on the nature of the cells, the humoral factor like erythropoietin or thrombopoietin, and the growth factors, the cell-cell interaction, and the pharmacologic substance like administration of specialized drugs which can inhibit bone marrow and inhibit erythropoiesis or megakaryopoiesis. Okay. So this is the stem cells. Okay. It go to division state. This is in stage 0. Then upon specific stimuli or growth factors, it gives rise to progenitor cells which can able to divide and proliferate, and this go to further division and then proliferate and differentiate into specific mature cells. At the end product, we will have millions of cells. Stem cell theory. So stem cell have the ability to, it is pluripotent. It gives rise to blood cells, which are able to proliferate, differentiate into mature cells. And these mature cells only normally release in the circulation. So during normal circumstance, I will not find any progenitor cells in the peripheral smear, on the peripheral circulation. So the one into passed in peripheral circulation and I can find in the complete blood picture is the mature blood cells. The immature blood cells I can find normally in the bone marrow sample. I will talk about erythropoiesis. So this was a general idea about hematopoiesis. So what about erythropoiesis? Erythropoiesis is the process with production of red blood cells. What are the stimulus? I mentioned before, decrease oxygen tension. If we remember, the function of the RBCs is oxygen carrying. They carry the oxygen from the lung to the tissue, and they carry waste product and carbon dioxide from the tissue to go out during expiration. So it is stimulated by decreased oxygen in circulation, which is detected in the kidney, which then secrete erythropoietin. This hormone stimulates proliferation. Proliferation means to divide, cell to divide and increase in size, in number, sorry. To divide and increase in number. Differentiate into red cell precursors, which activate increased erythropoiesis in the hematopoietic tissue, ultimately and lead to production of mature red blood cells. So in response to hypoxia or anemia or decreasing oxygen carrying capacity, the kidney release erythropoietin, which in turn lead to stem cells and commit stem cells to burst forming unit erythroid, and the end product will be mature red blood cells. So in the early fetus, this erythropoiesis takes place where? In the yolk sac. And by the third or fourth month, erythropoiesis move to the liver, as I mentioned in the early slides. After several months, the erythropoiesis takes place in bone marrow. Increase physical activity can cause increase in erythropoiesis. Okay? So physiological increase in activity can lead to increased erythropoiesis. In human, there are certain diseases is associated with increased erythropoiesis and lead to what is called extramedullary hematopoiesis or extramedullary erythropoiesis, which means that the liver and the spleen resume their original function and start to perform erythrocytes, as in a case of thalassemia and sickle cell anemia, which is a kind of hemolytic anemia. So the bone marrow, essentially all the bone produce the red blood cells until person is around the 5 years old. Tibia and femur is the site where there is hematopoiesis and erythropoiesis, and flat bone, the vertebrae, the sternum, pelvis, and ribs and cranial bone is the major site of erythropoiesis. So the definition of erythropoiesis is the production of RBCs, red blood cells, from the stem cells, regulated by what? By the effect under the influence of erythropoietin hormone. These pluripotent stem cells are committed to become a progenitor cells, give rise to erythropoiesis or red blood cells. It needs transcription factor. It's called GATA1. This transcription factor is important for erythropoiesis. After lineage commitment and hormones regulation, which is erythropoietin produced from the kidney, the cells take the lineage to pass into erythroid and mature RBCs. What are the erythropoietin? Erythropoietin is a glycoprotein released from a specialized cell in the peritubular capillary lining, the kidney. So it is specialized glycoprotein released from peritubular capillary lining, the kidney. Small amount of erythropoietin from hepatocyte can also form, but the major source is kidney. So decreased oxygen tension is the major stimulus for erythropoietin release. Erythropoietin bind to specific receptor in the surface of the marrow erythroid precursor, so it stimulates their division. So a common myeloid, so the stem cell are passed to the multipotent hematopoietic stem cell become myeloid progenitor. The multipotent stem cells after that become a unipotent stem cells. A unipotent, multipotent, can give rise to a several kind of cells. Then it commit to become a unipotent. Unipotent, it means that give only one kind of cells. So it become a unipotent cells. Then pronormoplast. Pronormoplast is the early detectable cell by morphology in the erythroid series in the bone marrow. It is also the other name called proerythroblast. These proerythroblast become basophilic or early normoblast, and then it become polychromatophilic or intermediate normoblast. Then this intermediate normoblast or erythrochromatic normoblast become late normoblast. I will see you now a figure. This late normoblast at this stage, all of these have nucleus. But here, the late normoblast, this is the last stage have nucleus, because at this stage the reticulocytes are formed because the nucleus is expelled. They are extrude the nucleus and they become reticulocyte. The reticulocyte is the immature RBCs that I can find in the peripheral circulation. This is the first stage I can see in the peripheral smear, the reticulocyte stage. Actually, the reticulocyte stage give me an image about bone marrow. If the bone marrow is active and work okay, or it is suppressed. Here, the hematocytoplast, as it's multipotent stem cells, which means that it can give lineage different kind of the cells. But when it become proerythroblast, that means that this is become unipotent. Unipotent, it give lineage only erythrocytes. So the proerythroblast become under erythropoietin, converted into and mature and proliferate into erythrocyte. How this happen? We'll see here. Okay, this is the bone marrow. Normal, I can find this red cell precursor in the bone marrow. Totally, I will not found any of this cell in the peripheral smear. If I found any of the precursor in the peripheral smear, it means that there is something wrong with the patient. So, stage of maturation of the erythroid series, proerythroblast or pronormoblast, the other name. Then it become basophilic normoblast. Basophilic, which means that the cytoplasm is blue in color, because this cell does not have hemoglobin yet. The red color of the erythrocyte are from the hemoglobin content. Then polychromatophilic normoblast, and then orthochromatophilic, which means that the color is red in color. As I mentioned here, there is the cell are large size, the blood cell are large size, it becomes decrease in size, and till in the stage of orthochromatophilic normoblast, we have a nucleus here. This nucleus will be extruded and it gives us to reticulocyte stage. The reticulocyte stage normally found in the peripheral smear, but it is stained by special stain which is called supravital stain because it have a remnant of RNA. Erythrocytes have no nucleus, so I have to know that these two cells are the only detected cells in the peripheral smear. This I can detect them on the bone marrow smear. Again, a very nice picture to show how is the nucleus extruded. Pro-erythroblast, here this is the stem cells or committed stem cells, become pro-erythroblast, early erythroblast, and intermediate, and then the late normoblast, which have eccentric nucleus. This eccentric nucleus will become extruded and give rise to reticulocytes. Only the reticulocytes are formed from 0.5 to 2.5% of the total erythrocyte mass in our body in peripheral circulation. If reticulocyte increase, it is increased in some special situation, like after hemorrhage or hemolysis, or after treatment of anemia. And the mature RBCs, as I mentioned before, non-nucleated cells or enucleated cells with a diameter of 6 to 8 micrometer, but actually it is around 8. So the cell released from the bone marrow in the reticulocyte stage and circulate in the range from 0.5 to 2.5%. So what are the essential elements for erythropoiesis? We need iron, we need vitamins, vitamin B12, we need vitamin B6, and we need folic acid and amino acid. Lack of any of these essential element will lead to any form of anemia. We'll discuss about this form later. This is the picture of mature red blood. This is the picture of reticulocyte. The immature RBCs, okay, the one which have a reticular fibers, which is a remnant of RNA. This is stained by supravital stain, and this is the mature RBCs. It doesn't have reticular fibers. Of course, this figure from one which have increased hemolysis of RBCs and lead to a stimulation of bone marrow and erythropoiesis. So, as I mentioned, reticulocytes are immature RBCs present normally from 0.5 to 2.5% of red blood cells, and it does not stained with normal stain. It is stained by supravital stain like new muslin blue or brilliant chrysal blue. This is the mature red blood cells. The normal red blood cells have the red color due to the hemoglobin content, and this red blood cells does not have nucleus. It is non-nucleated cells. Discoid shape and have central pallor not more than 1/3. It have central pallor. It is of diameter of 7 to 8 micron in diameter. They have a central senar region, okay, viewed by microscopically as a lighter central area, the central pallor. This is the red cells. This is another picture of reticulocytes, and this is by brilliant chrysal blue stain, and this is the normal RBCs. Okay. So what are lead to increased erythrocyte or erythropoiesis in case of severe blood loss, hemorrhage, trauma, accident, or anemia and decreased oxygen carrying capacity? We'll discuss all of this later on inshallah in the other lecture. يبقى احنا بنلخص اللي احنا بنقوله دوت ان شاء الله في ان احنا خدنا النهارده الهيماتوبييسيس ان جنرال، وعملنا فوكس على الاريثروبييسيس. وقلنا ان انا عندي البلاد متكون من سيلر كومبوننت ومن بلازما كومبوننت. وقلنا ان البلازما ديت فيها نيوترانت، فيها الفيتامينز، فيها امينو اسيدز، فيها بروتين، فيها كواجليشن فاكتورز. وقلنا ان انا عندي السيلر كومبوننت بتوعي عباره عن اريثروسايت اللي هم ار بي سيز وميجا كاروسايت بليتلت ووايت بي سيز. وقلنا الوايت بلاد سيلز ار ريسبونسبل فور ديفense ميكانيزم وبتلعب دور مهم قوي في الاميونيتي. والار بي سيز مهمه جدا، الريد سيل مهمه جدا في انها الاكسجين كارين كاباسيتي. وقلنا ان البلاد فورمينج اليمنت المادر سيلز او البريميتيف سيلز هي عباره عن الستيم سيلز. وقلنا الستيم سيلز ليها ability of self renewal و proliferation and differentiate. هي undifferentiated cell resemble a small lymphocytes but have the ability to be self renewal to maintain the stem cell pool and differentiate into specialized cell line upon to specific growth factors. What are these growth factors? It may be cytokines or hormones. Hormones like thrombopoietin or erythropoietin, and growth factor like and cytokines like interleukin 3. As I mentioned before, the interleukin 3 is pan-hematopoietic growth factor. It stimulates hematopoiesis in general. I want to show this figure. Okay, and we have a stem cell factor. Normally, these stem cells are present in stage 0, G0. Upon specific stimuli, it can divide and differentiate and proliferate into specified cells. If they are exposed to, if there is a condition of allergic condition or parasitic infestation, they will be